Hello, and welcome to the CAP Today webinar for Tuesday, April 22nd. I'm Bob McGonigal, the publisher of the CAP Today, and I'll be your host, and I'll moderate a question and answer session after our formal presentations today. Today's webinar is entitled Harnessing the Power of Microbial Cell-Free DNA, Practical Insights for Implementing Metagenomic Sequencing in Your Lab. This very important webinar is made possible by a special educational grant from Karius. Karius is a CLIA certified and CAP accredited laboratory with headquarters in Redwood City, California. We'll feature a single speaker today, whose picture you've seen, Dr. John Farrell. He's a ID physician, the director of Microbiology and Serology Laboratories, and the chair and medical director of infection control and prevention at OSF St. Francis Medical Center here in Illinois. We also have a special guest who I will mention and introduce in just a moment. But first, I wanted to go over a couple of housekeeping items with you. You'll see a Q&A pod on your screen at any time during today's session. You can make your comments and ask your questions. As I said, we'll deal with those in full after our formal presentations. Also, if you have any technical issue, you can type that in. We have live operators standing by to help you with any technical issues. We do plan to post this webinar with all the audio and slides in about a week. So stay tuned for that. It'll be on captodayonline.com. You can expect some follow-up email both from us and from our great sponsor, Karius. And finally, I do need to let you know that CAP Today does not endorse any products or services that may be mentioned today. Also, any remarks of mine are purely personal, not to be taken as policy of the College of American Pathologists, as is obvious. But now let me introduce to you our first speaker and my surprise guest, and that is Dr. Tim Blauwkamp. Dr. Blauwkamp is a PhD biochemist, and he got that degree at the University of Michigan, not far from Chicago, the Harvard of the Midwest, as we often refer to it. But he's a very experienced executive in this world. He did a postdoc at Stanford and has been involved with advanced diagnostics for most of his career. And with that, I'm going to ask Dr. Blauwkamp to begin his presentation. Dr. Blauwkamp.
Well, thank you for that, Bob. And good morning, everyone. Thank you for joining us today as we explore the power of microbial cell-free DNA and specifically how to harness that power toward better infectious disease diagnostics. Now, my role today is very limited. I have the honor of introducing the technology background over the course of the next 8 or 9 slides or so, prior to handing it over to Dr. Farrell to describe his experience using this technology at OSF. And so with that, I will begin. The idea of using metagenomic sequencing to help with infectious disease has been around for a while and is definitely gaining attraction and attention lately. There's a few different flavors of how one might approach using DNA sequencing for infectious diseases, ranging from ribosomal amplicon sequencing to sequencing of microbes found in the infected body fluids. Today, I'm going to focus on the approach that Karius has developed, and that is to sequence microbial cell-free DNA found in plasma. And this approach is based on the observation that the microbes throughout our body, and particularly those causing infectious disease, they don't live forever. They turn over, and when they do, they release fragments of their unique genome into the bloodstream to be recycled or excreted like all the other nutrients and waste in our body. Now, Karius has developed extremely efficient methods of capturing the microbial cell-free DNA from the bloodstream and converting that into NGS sequencing libraries, and ultimately using that to identify which microbes are contributing abnormal amounts of cell-free DNA. And it doesn't require the microbe itself to be in plasma. And so for those of you familiar with other cell-free DNA-based diagnostics, particularly human cell-free DNA-based diagnostics like those used for non-invasive prenatal testing or used for solid organ transplant rejection monitoring, and certainly those used for tumor profiling from plasma or minimal residual disease monitoring in the cancer space. The easiest way to think about the approach that Karius developed is as a liquid biopsy for infectious disease. The technical challenge is alluded to here in the bottom right, where you see the relative amount of your DNA of interest against the background of other types of cell-free DNA that are present in the plasma. And in the case of microbial cell-free DNA, there's typically about a millionfold excess of human DNA relative to the signal. But we know that there's an enormous amount of total DNA in the blood now, and more than enough to use the microbial components if you can fish them out as the basis for infectious disease insights. And so the value of taking an approach like this is analogous to the value you might see in the other cell-free based diagnostic applications. The first one being that you don't actually need to isolate the organism of interest from a body fluid or a tissue or the blood itself, because you can see the cell-free DNA signatures in the plasma, even when the microbe itself is not. And you really see those signatures from anywhere that microbes are growing that have access to the bloodstream. And we have a number of papers showing the ability to see those localized infections despite the absence of detectable intact pathogen in the bloodstream. So that's the value of the cell-free DNA component. But if you do it if you do the sequencing in a way that's totally non-targeted, you also have the ability to test for every known microbe or every microbe with a reliable reference genome sequence simultaneously without having to develop any specific chemistries that are targeted towards one class of microbes or one class of microbes or another. And so the approach that we've taken is to focus on removing the human DNA from the plasma and sequence everything that's left such that we can see anything with a high enough quality reference genome. And we can rapidly adapt the database as emerging pathogens appear. And finally, the other just element of value that goes a little bit beyond maybe some of the other diagnostic areas is that we've developed the control strategies to take advantage of sequencing as a digital readout, one that's highly amenable to accurate and precise counting algorithms, such that in combination with the internal controls that I'll just briefly touch on, we can provide an absolute quantitation of the cell-free DNA concentration for each species that's observed in the sequencing data, enabling an ability to monitor the change in that plasma cell-free DNA sequencing over time with repeat testing. And so, this slide summarizes the value, very analogous in many cases to the value we've seen in other cell-free DNA-based diagnostic applications. But I also want to double click on the challenges that become just as obvious as the value as soon as you dive into actually doing this. And so this image represents one of the first experiments that we did to assess off-the-shelf capabilities of the best available laboratory kits for extracting microbial cell-free DNA and converting it into libraries, as well as the best available compute pipelines for analyzing that data. And what we found through this study of 20 specimens, 10 on the left with confirmed culture positive infections and 10 on the right from essentially healthy individuals, is that there's a huge signal to noise challenge, particularly with microbial cell-free DNA, where the signal we're interested in is highlighted in these orange dots. That's the concentration of the microbe cultured out of the blood specimen. And all of the blue dots are false positives from a variety of different sources. And so the challenge is, of course, that while you can, from a proof of concept or feasibility or even research perspective, often see the signal you're looking for if you know what to look for, more often than not, that signal is buried deep in the noise, or in many cases, not even visible because of this high human DNA background. And that's in patients that we know have infection, but the challenge is just as severe in healthy patients, making it really challenging to get the utility out of an approach like this with off-the-shelf tools. And so the first couple years of Karius's existence were dedicated to identifying the source of these signal-to-noise challenges, which are shown here on the left, and then ultimately trying to address them in ways that makes this usable and useful at scale. And so I think many people who study this now are very familiar with these types of challenges, but I'll just mention them. I mentioned the high human DNA background, roughly a million-fold excess. One of the most difficult challenges is this environmental contamination. All of the reagents and surfaces that are going to touch the plasma specimen have fragments of microbial DNA that get mixed in with this endogenous signal derived from the plasma. And it gets challenging to sort out which came from the reagents or plastic surfaces and which came from the plasma itself. And so in many cases, that environmental contamination signal will even be much larger than the signal from the pathogen. So we spend a lot of time solving that. And then, of course, there's all the compute challenges associated with accuracy of the reference databases, the diversity of microbes that are detected, which range in GC content from 20% to 70%, and often have biases in efficiency for recovering those different GC contents, as well as the fact that we're never studying a microbe in isolation when you're surveying the body. It's always a mixture of dozens to hundreds of species, and those genomes share significant portions of sequence. And we want to develop the compute algorithms to accurately dissect which microbes are contributing to that mixed population of signal. So, of course, we wouldn't be talking today if we hadn't actually done all of that. And as part of that very early proof of principle, we applied the methods to the same specimens on the left, and of course showed that we can pretty dramatically increase the signal and pretty dramatically reduce the noise in both the patients that are known to have an infection and, importantly, in the patients that were healthy. It's also important to note that it's not a perfect process, and there's been dozens of releases over the course of the last 10 years or so that we've been doing this. But there will always be an element of challenge to figuring out exactly what's in the specimen and what's causing disease from that specimen relative to other sources of information that's less meaningful. Once we address the other thing that I just want to double click on is it's one thing to address the errors in the accuracy per specimen, but this is a really powerful and highly sensitive approach that faces some of the same challenges as other very sensitive approaches like quantitative PCR. And so when we started the operationalization of this, we really learned as much as we could from those other high throughput labs doing very sensitive testing at scale, and developed a number of quality control strategies that we believe address the unique challenges of metagenomics. We can go through these in more detail. They've also been published in Nature Microbiology back in 2019 and several publications since. But I'll just say that some of the key elements of that include real-time monitoring of that environmental contamination background by using no template controls that match the physiological characteristics of your specimen. And so you don't want to use you don't want to use buffer to begin with, because that doesn't mimic the recovery profiles you get from plasma or other body fluids at all. And you want to tailor that matrix, whatever you're using to monitor that background, to something that matches the background of your specimen type, because you'll get a very different view of that background if you're not matching that no template control matrix to your testing matrix. The other elements I'll mention is just with a sensitive approach, you want to do things like control for exquisitely rare events. Things like carryover and cross-contamination could easily mislead you when you run a bunch of samples in batch. Things like differences in the GC bias or the fragment length bias as you're running a protocol could easily lead to differences in sensitivity for your GC-rich or AT-rich microbes on a day-to-day basis. And things like even just as simple as mixing up samples when you're running them through thousands at a time can lead to mix-up. So we have these controls that are meant to mitigate the risk associated with each of those previously observed failure modes. And on the right, of course, we quantified the risk without any of these measures, and then, of course, reduced the risk of false positives being derived from these operational risks about 1,000-fold through these controls. And the end result of all of that then is this process that we have referred to for most of our history as the Karius test. It's recently been renamed the Karius Spectrum send-out test to distinguish it from a BAL test that we're launching here in the next couple of weeks. The way this test works is it starts with a standard 5 ml collection of blood into ideally a plasma preparation tube. That tube is centrifuged at the ordering institution and then shipped at ambient temperature to Karius in the packaging that's provided. We go through an automated process once those specimens arrive every morning about 8 a.m. Here in Redwood City. There's a largely automated process that takes about 6 hours to go from the time we load the plasma to the time that we have sequencing ready libraries from that entire batch. We sequence those overnight, and when the sequencing is complete, we have a computational pipeline that automatically picks up the data, compares it to our reference database, figures out which microbes are derived from the plasma and what the relative abundance of all those microbes derived from the plasma are, and then compares those abundances to a variety of both real-time controls, like that environmental background control, but also historical controls that characterize the normal variation of every one of those microbes in healthy populations, such that the final report only contains microbes that exceed the bounds of normal variation in a healthy human population, and which pass a number of analytical controls, including being extremely unlikely to have been derived from any of the contaminating sources. I know Dr. Farrell will go through some of the report elements, so I won't mention it here other than to say we're constantly doing our best to make this as simple as possible, listing only the microbes that are observed beyond the bounds of normal human variation and listing the exact absolute quantitation of that microbe cell-free DNA in plasma in units that represent the number of unique fragments from that species in 100 nanoliters of plasma. For those that are less familiar with the test at scale, we published a couple years ago an analysis of just what the standard testing process looked like over more than 18,000 samples. And it's surprising to some that are less familiar or have experience with earlier versions of metagenomic testing that about 40% of the tests we report back have no pathogens identified that exceed all of the analytical and clinical filters that are applied prior to reporting. And so there may be dozens or hundreds of microbes, even microbes derived from plasma, microbial cell-free DNA species derived from plasma, but they don't exceed those bounds of normal variation. Among the 60% of reports that are positive, about half of them have only a single pathogen, and then there's roughly a twofold decrease in the number of reports with 2 or 3 or 4 pathogens. And not surprisingly, if we look at the superkingdom distribution, most of the microbes on the positive reports are bacterium, with about 20% viruses, 8% fungi, and some very impactful parasitic infections identified as well. Operationally, we strive to make sure that there's a high success rate. I'll just double click a couple of things here. We have about 97.3% of the reports being returned within 24 hours of our receipt of those reports. And so if you're sending those specimens directly to Redwood City and the packaging that's provided, across the nation, it takes a median of 24 hours for those samples to get here, and then you get the results within 24 hours of our receipt. If you're going through a different reference lab, that usually adds 2 to 4 days to the time it takes to get to Karius. But once they get here, you get that same within 24-hour turnaround time of these results. And finally, on my last slide, There's a lot of discussion today, rightly so, about how to deploy a technology like this in the most diagnostically responsible way. There's more than 200 publications by now on how the Karius test has been deployed at various customer sites. If there's a patient population of interest or a use case of interest, there's probably at least one case series or even study on it. This just represents some of the most clinically impactful use cases, as well as some of the references associated with how it's been deployed across these potential use cases. And with that, it's my honor to complete my introduction before handing the microphone back to Bob to introduce one of the most experienced users of the Karius test to talk about his journey implementing this technology at OSF. And so, Bob, that completes my introduction. Thank you.
Dr. Blauwkamp, thank you very much. What a wonderful presentation that you've given us. I had really no idea that the level of sophistication that a test like this would allow, and also clearly clinically convenient in ways that one might not have imagined in terms of turnaround and dealing with what seemed to be quite clear reports. But for the complete experience on the clinical side now, it's my great pleasure to introduce Dr. John Farrell. He's professor of medicine at the University of Illinois College of Medicine. As I said before, he's an infectious disease doctor, medical director at the OSF System Microbiology, Immunology, and Molecular Diagnostics Labs. He's very experienced, and as we were chatting before the lecture, he's done everything from soup to nuts, and I think still does. So he's a great laboratorian to tell you about the OSF experience with the Karius test. Go ahead, Dr. Farrell.
Thanks, Bob. I sure do still practice soup to nuts, and that's the exact expression I tend to lean on. Thanks for the intro, Tim. Appreciate it. Happy Lab Week, everyone. For those of you who aren't familiar with this healthcare system in the Midwest, OSF stands for Order of St. Francis, which was founded in 1875 by the Third Order of nuns who emigrated from Germany to the U.S. They opened their first hospital a year later in 1876 in Peoria, Illinois, by converting a house into a 32-bed hospital, which has evolved to today a 670-bed teaching hospital with residency programs in internal medicine, emergency medicine, pediatrics, obstetrics and gynecology, radiology, general surgery, neurosurgery, fellowships in interventional radiology, gastroenterology, pulmonology, critical care medicine, just to name a few. The hospital in Peoria remains the heart and hub of the organization. There are 25 hospitals across the system with over 700 provider offices spread throughout Illinois, Wisconsin, and Upper Michigan. But other than the most basic testing performed at outlying hospitals, our lab serves as the reference lab for all the hospitals in the system, as well as other hospitals in the region. We have a completely automated walk-away WASP system in microbiology. Our serologies and chemistries are automated on the Abbott Architect Tract. And we're an Epic Beaker shop. Epic order entry and integrated EMR on the patient and provider side. Beaker lab resulting reporting and IT interfaced on the lab side. Karius test is orderable through our Epic order entry system. We originally contracted with Karius as a direct send out test for our hospital in 2007. Currently, there are only 2 hospitals in the system where a Karius test is orderable on inpatients. And not coincidentally, these are the 2 hospitals that are staffed on site by UIC affiliated ID physicians. We partner with the ID providers in this group to provide stewardship for this test. And we feel that that experience has been value added. Certainly for labs such as ourselves that were Becton-Dixon-based labs that relied on BD, blood culture, liquid media. You all can relate to our experiences last fall when we almost ran out of blood culture bottles. So the tip of the iceberg in stewardship is screening that's hard baked into the EMR. Our Epic order entry system mandates for any new patient, first set of blood cultures has to document appropriate clinical indication in the order and in the EMR. Likewise, for follow-up or serial tests, appropriate clinical indication for documentation of clearance of bacteremia must be documented. Certainly that allowed us to get through that shortage by the skin of our teeth without running out of blood culture bottles. But that's really just the tip of the iceberg of stewardship for limited laboratory resources. That's why we feel our partnership with the ID providers has really been value added. Here's an example of, I went into a patient's chart last week and ordered 2 sets of blood cultures as well as a Karius test. As you can see, the EMR immediately reminds me the patient already had 2 sets of blood cultures done 4 days prior that were negative and prompts me for the option to remove these redundant tests. Also reminds me that if I wasn't an ID provider, the Karius test would not be available in my order entry menu. We feel that our track record suggests this partnership has been a successful example of stewardship of the test. So just looking at a 16-month snapshot of our experience dating back to Jan 1, 2024, the 344 tests over that time averages 5 tests a week. So we're averaging 1 test a day on Monday through Fridays. There are select clinical situations where a follow-up test, the analogous situation of documenting clearance of bacteremia, is appropriate, but that's only 10% of the time in our experience. And I'm going to just share a couple quick cases that demonstrate the value of follow-up testing. This patient presented to our hospital in November of 2024. He was brought in by his family. If I remember correctly, his weight in the emergency department was 95 pounds. He was an end-stage AIDS patient with CD4 count of 4, and he was confused, disoriented. Based on his obtunded mental status, he did have diagnostic imaging, both CT scan of the head and gadolinium enhanced MRI of the brain, neither of which showed any evidence of CNS toxoplasmosis. His chest x-ray had scattered nodular lesions that could have been anything in the differential, was certainly broad and included pneumocystis and tuberculosis. He was admitted to the hospital, placed in isolation. 2 days later, his original Karius test returned with a surprising revelation that he had very high levels of Toxoplasma gondii, 19,000 molecules per microliter, which is extraordinarily high. And his pulmonary infection was actually pulmonary histoplasmosis with 2,200 MPM as well. So he was started on highly active antiretroviral therapy at admission, as well as twice a day, 300 milligrams itraconazole for pulmonary histoplasmosis and pyrimethamine, sulfadiazine, leucovorin, triple drug cocktail for Toxoplasma gondii. Given that he was as malnourished as he was, and there was not much clarity and certainly clinical concern about his ability to absorb and metabolize this high pill burden, a follow-up test was performed, and that is the original test I showed here with the much lower levels, almost 2 log decrease in toxoplasma and a 1 log decrease in histoplasma. The Karius test does include this information on page 2, showing obvious trends one way or the other, in this case, obvious trend toward improvement. Second case worth sharing briefly was a gentleman who was almost a doppelganger of this first patient. He was brought into the emergency department by his family as well, his wife actually, with fevers, chills, night sweats, cough. He was a gentleman who actually weighed north of 100 pounds, but not by much. He had a history of esophageal cancer of the lower esophagus. He was status post resection and anastomosis, as well as chemotherapy and radiation therapy, also quite malnourished. His chest x-ray showed cavitary infiltrates, again, a differential quite broad, including invasive fungal infections and TB. And again, he was placed in isolation. 2 days later, the Karius test came back, demonstrating his pulmonary infection appeared to be a Mycobacterium abscessus infection. And although this organism is admittedly a rapid grower, providers did not feel that it was reasonable to wait for culture confirmation and then susceptibility testing, which we send out to National Jewish in Denver. And he was started on empirically on treatment. Follow-up test was performed 2 weeks later, and the M. abscessus was undetectable, suggesting he was not infected with a multi-drug resistant strain of Mycobacterium. Speaking of drug resistance, the Karius test does detect multi-drug resistant organisms, aka MDROs, including VRE, MRSA, ESBL producing gram-negative bacteria, as well as carbapenem resistant Enterobacteriaceae, commonly referred to as CRE. I thought when I was making the slide deck last week, it might be informative just to take a couple screenshots of patients from the prior week and let you see how these results flow into the EMR. So we have a couple cases here where you can see the test was ordered, collected, centrifuged, and shipped via FedEx to the Karius lab on April 4th. Less than 48 hours later, we had a result in the EMR without any indication for addending the report with antimicrobial resistance results. When the detection is an organism that could potentially be associated with antimicrobial resistance, the initial report typically appears in less than 48 hours, and then an addendum will appear 24 to 48 hours later with information based on detection or lack thereof of genetic markers of resistance. So if we just look at these top 3 patients from the week prior, when I took this snapshot, you can see patient number 3 from the top had an organism detection in less than 48 hours. And there's a footnote on the status of the report indicating that this organism does warrant screening for genetic markers of resistance, and that result will be forthcoming in 24 hours. That patient actually had very high level of Salmonella enterica, which did grow in the blood as well. 28,000 almost MPM is extremely high. He was septic and quite ill. The following day, the screen for CTXM, the most common community-acquired genetic marker for ESBL, was reported as negative. The patient above, where we had a turnaround time in less than 48 hours, was a negative detection, as Tim alluded to. If there are no microbes detected other than background, this is the result you'll get in less than 48 hours. And then this top patient on this screen was a patient who had Enterococcus fecalis detected in less than 48 hours. And then the VAN A, VAN B screen came back the following day. So ultimately, complete information was obtained in less than 72 hours. When I scrolled back the week prior, took another screenshot. I thought it was worth sharing that screenshot since we see 3 examples in that 14 or 15 patients where the result is flagged with the red alert. And I think for laboratorians, it's important to point out that in our lab, this is not treated as a critical value. Our lab is not making phone calls on these results. That would actually be redundant given that the ordering provider, of which there are only the ID physicians in the UIC group, is already received a text to their phone notifying them a result's available, and they can query the result on their phone via the secure Karius app any time of day or night. So the ID providers do appreciate not being called in the middle of the night. I can trust me, one of the reasons they went into ID is so they can avoid getting woken up in the middle of the night. The um, the lab certainly appreciates not having to make those phone calls, since that's the biggest impact on lab productivity is sitting on hold. So avoiding that is very impactful for the lab as well. I think it might be worth just taking a quick look at these 3 cases. The first patient, there are several results of viruses, Candida, um, kind of normal oral flora, and viruses you might expect to react in an immunocompromised patient. And you'd be right, this was an immunocompromised patient. This is a gentleman who had CLL, who had completed induction therapy and then was readmitted 3 weeks later. Febrile neutropenic chest x-ray showed what appeared to be a necrotizing pneumonia, and certainly the detection of Aspergillus fumigatus, which did not grow in his sputum cultures, was impactful. He did respond to treatment with voriconazole. This second patient was an immunocompetent patient, a gentleman in his 50s who had bilateral pulmonary nodules. So he had a low level detection of histoplasma capsulatum, which is fairly typical given that this is an intracellular pathogen that provokes very prominent granuloma formation in the lungs. He was negative by urine antigen, negative by immunofussion with no H-band, no M-band, but he did respond nicely to itraconazole therapy. And the final patient at the bottom of the screen was another end-stage AIDS patient. This gentleman had a CD4 count of 7, and no surprise to see pneumocystis detection. He did have an abnormal chest x-ray and some shortage of breath, but the finding of Mycobacterium avium intracellulare complex was not expected. Not surprising, certainly, given the CD4 7. Anyone with a CD4 count under 50 is at risk of MAC, but certainly impacted his management. The treatment of pneumocystis usually involves TMP, SMX, and prednisone, but prednisone was held given the disseminated MAC infection, which was treated with a drug cocktail to which he responded. At our hospital, pneumocystis has been and continues to be the most common opportunistic pathogen identified over the same 16-month snapshot dating back to Jan 1, 2024. We see pneumocystis far outpaces any other pathogens in our experience. Second is, or runner-up, is Histoplasma capsulatum. Not surprising given our location between the Mississippi River and Ohio River. We have several detections of non-tuberculous mycobacteria, NTM, including MAC and M. abscessus case that we looked at. Since we're not a stem cell transplant center, although we do have an oncology ward and we do have a solid organ transplant ward, we don't see a high number of cases of invasive fungal infections. Given our experience with pneumocystis, it's not surprising that our ID group did publish a retrospective series of 23 cases. Um, I think what's really interesting to me about the publication is for somebody like me who cut my teeth taking care of AIDS patients in the era when antiretroviral therapy didn't exist, and in the early days when it did exist, every patient with suspected pneumocystis required interrogation of bronchoalveolar lavage fluid to look for the, at the time, pneumocystis carinii, now pneumocystis jirovecii, under the microscope. Um, this population, 61% of the patients never even went to the interventional pulmonary lab for bronchoscopy. That's really impactful to the bottom line for an organization such as ours, a charity hospital that takes all comers. Obviously, AIDS patients are a marginalized population. Typically, reimbursement for inpatient care is funneled through the Center for Medicaid and Medicare Services, and that's based entirely on diagnostic related groups that are coded when the reimbursement is submitted. And now I'm probably getting out of my lane when I step into healthcare economics. It's complicated, but I am confident that the bottom line is improved if we can avoid tying up in a lab or an OR with interventional pulmonologists, a staff from anesthesia, as well as staff from the path lab. And given the fact that we typically have pneumocystis case in-house at any given time, that's a savings of millions of dollars to the organization every year. That publication was uh, in 2022 at the time when we started to see much more interest specifically in the Karius test. Um, the number of publications citing Karius specifically has grown exponentially over the last 3 to 4 years, as you can see from PubMed. There have been a number of meta-analyses and systematic reviews of the power and the impact of microbial cell-free DNA sequencing. One common theme that has emerged from this literature is the impact this has for immunocompromised patients, where time is of the essence. So patients with febrile neutropenia, patients with pneumonia, particularly necrotizing or cavitary pneumonias in oncology patients, solid organ transplant patients. This rapid recognition of fungal infections that often would require weeks to cultivate on SAB high or PDA augers in the lab is a sea change. Culture negative endocarditis is a syndrome also commonly found to benefit from more detections, more sensitive detections with this technology. Um, in our center, where we struggle sometimes to get biopsies of vertebral bodies or disc spaces, we have found some benefit as well in discitis and vertebral osteomyelitis. But I'd say the literature seems to coalesce around fever of unknown origin as being the syndrome where this test is highly impactful. So fever of unknown origin, aka FUO, is defined as temperature, objective documented temperatures of greater than 38.3, 100.9 Fahrenheit for 3 weeks with no clear explanation. So 2 months ago, in February of 2025, group at Mayo published their experience with the Karius test specifically for patients with FUO over a 4-year period. This is a worthwhile read, in my opinion, and certainly available on PubMed. Although if you just Google chart biopsy to liquid biopsy, this paper will be your first hit. They stratified their testing for these patients into 2 tiers. Their cohort was 176 patients who met the criteria for FUO. Only 16 of those patients actually had an explanation diagnosis based on simple tier 1 testing, hepatitis and HIV serology, syphilis serology, or blood cultures. Tier 2 testing comprised screening for culture negative endocarditis with serologies for Coxiella, Bartonella, Brucella, as well as screening for tick-borne infections like Rocky Mountain spotted fever, Ehrlichia, Anaplasma, Babesia, Lyme disease, as well as the huge for fungal diagnostics, the histoblasto-urine antigen we're all familiar with, as well as Aspergillus serum antigens and the beta-1,3-D glucan, aka Fungitel test that is ordered so routinely. And they found the yield, diagnostic yield for those tier 2 tests was 7%, 4%, and 6% respectively. Ultimately, they found a third, specifically 69 of the 176 patients had a infectious disease that was the source of their fever. And when they extrapolated those 69 patients into this 4-colored bagel here on the right, they found Karius test, that was the platform they used for metagenomic sequencing of microbial cell-free DNA and plasma, agreed with tier 1 and tier 2 testing in 21 patients. There was partial agreement, but some discrepancies in 7 patients. And then the Karius test was the only diagnostic platform that was diagnostic of the true cause of fever in these patients with infection in 15 patients in that cohort. So 2/3 of their patients, they were basically ultimately diagnosed by microbial cell-free DNA sequencing. This experience agrees with our experience. We have also found in the hands of our infectious disease providers that our Karius tests come back positive 66 or 2/3 of the time. Organisms most commonly reported are enterogram negatives, E. coli, Klebsiella, enterogram positives, both Enterococcus faecalis and Faecium. The number one bacteria detected over this snapshot that we've been looking at beginning Jan 1, 2024 is Pseudomonas aeruginosa. The top 6 viruses detected are all herpes viruses, all from the herpes family. And then the fungus most commonly detected are oral flora that found in the oropharynx Candida species Saccharomyces. So clearly these detections require a intimate understanding of the patient context, which is of course why we have found that this partnership with our our UICID group has been a win-win. Certainly this test is a different animal than sending samples out to our core sequencing facility where we would get sequences back, but not in 2 days. And we would then be responsible for the bioinformatic interpretation. Obviously, as you saw, having the organism detection to the species level in less than 48 hours is obviously impactful. We certainly in the lab appreciate that it's integrated into the EMR, saving us on phone calls. The providers obviously appreciate that as well, not to mention the fact that they can lean into the genetic detection of antimicrobial resistance markers to guide therapy. We feel that this test actually has an excellent return on investment, given that we're able to avoid invasive procedures in patients who otherwise may be languishing in the hospital without a definitive diagnosis. And when you think about it, infectious disease providers are not interventionalists. They don't perform endoscopies. They don't do surgeries, take patients to the OR for invasive biopsies. They, what they bring to bear when they're consulted to see a patient in the hospital is an attention to detail, a understanding of the clinical context and the patient's immune status. And we find, really appreciate the advantage of having metagenomic sequencing of microbial cell-free DNA plasma in their toolbox. To have that liquid biopsy in their toolbox is value added for them when they're called to see the patient. Certainly, this is the medicine of the future, but I would say the future is here now. So I'd like to thank Travis, our administrative lab director, Mike, the president of our PATH group, Heather and Jen, the managers of microbiology and the send out lab, respectively. Annette and Nick are send outologists, Shayla, Bailey, Tobias, Tamara, Levi, Ross, Anderson, just to name a few, are processingologists. I also would be remiss if I didn't mention our CLS students who are tuned into the webinar, as well as Doug, the chair of the UIC group that I've mentioned in this talk, and Dr. Prokop would wring my neck if I didn't thank him for his mentorship in my micro training at CCF. I'll turn it back over to you, Bob, to monitor the questions.
Thank you. Thank you so much.
My pleasure.
That was an incredibly interesting talk, and I know we're going to have some questions coming up very quickly. I'm going to try to turn my camera on for us all. There we are. And in particular, we've heard two very rich presentations about this test and its use. I'm going to address my first question to Tim, and I want to let everyone know that you can ask questions and make comments at any time. Just type it in the pod there for Q&A. They'll come up and we'll ask them in turn. But I want to start with you, Tim. Clearly, you've engineered some important abilities for systems and labs to have great stewardship over this kind of test. Was that engineered from the very beginning and recognizing the importance of having some restrictions on ordering?
It's a great question. In fact, the things we spend most of our time thinking about evolve over the course of the company, and actually how we help physicians and labs with that stewardship question is one of the top things we think about every day right now. So the short answer is, earlier on, of course, that wasn't our top priority. We were thinking about how do we get this analytically accurate. And then you evolve through phases of getting it clinically valid, where we introduce a lot of these clinical filters that, capture the normal variation of human background signal or human microbiome signal. How we narrow or adjust the microbes that are reportable to match what's clinically relevant. The efforts towards stewardship that we take on now have to do with just continually investing in that what's called a clinical utilization review that Dr. Farrell showed a couple of times. It's automatically generated, I think, either monthly or quarterly. Lorraine can help us on that cadence, but it's an unbiased look at every single test and result that an institution has sent, provided back to the lab specifically to keep the laboratory leadership aware of how the test is being deployed within their institution and what the results look like from a positivity rate, from a turnaround time, from an antimicrobial resistance marker detection capability. The other side of that coin has to do with how it's being clinically deployed. And there, we rely on the experts. And so we provide as much transparency into what institutions are taking this seriously and developing algorithms for specific types of use cases. We provide as much help as they want or as we can to publish that, just as a way to get what's working in the community more visible to the entire community.
Thank you.
One of our top priorities right now is helping on that front.
Thank you very much. And of course, Dr. Farrell, there'll be a lot of questions to you about this very same issue. So we have one from a participant who asks, do you actually have a formal send out approval process within OSF before you send out samples?
Great question. And when we originally partnered with Karius, we debated about that. I think we came down on the right side when we decided against that. Um, as I tried to explain, I think there's real investment in the ID providers in using this test judiciously with discretion, because they do value having it in their tool bag, as I mentioned. And so they want to be confident that they'll have this available going forward. So they're completely invested in appropriate judicious use of the test. We do not uh, block their orders, screen, and then approve. We do the reviews quarterly. We meet with their group quarterly, and it's been a win-win.
Very good. But just to follow up as the medical director, we all know that there's a lot of very high profile, highly conscious infectious diseases. Sepsis, of course, is kind of a holy grail of concern everywhere you go. Do you yourself find a lot of need to answer phones to explain what the Karius test can do and what it can't do, and to help people triage those kinds of decisions before they get to the ID docs?
Uh, well, I do get phone calls for sure, Bob. The typical phone call is from a pulmonary doc or a gastroenterologist
Right.
who wants to get the Karius through the back door.
I
Um,
understand. Yes.
and that's easy for me to handle. I just let them know that the infectious disease providers are available for consultation, and that's what I lean into.
Very good. Very good. Tell me just a little bit about now introducing this resource to a large healthcare system such as the one you're with. Did you have some formal lectures, some Zoom meetings, uh, a
Yes,
newsletter? Just tell me how you went about it. I'm sure we have many people who be interested in that.
all of the above. I presented grand rounds, did several morning report and noon conferences in multiple disciplines, pediatrics, as well as medicine, as well as just meeting down in the ED with providers. Um, the uh, uh, I also, at this point, feel like I've handed the baton to Doug's group and let them take it from there. Ever since they began publishing their experience, I feel like it's they're invested in educating our healthcare organization, because to be honest with you, they're invested in expanding their group's reach. They
Of course.
aren't they aren't actually officially involved in care for all the patients. They try and expand their reach through telehealth, but by virtue of educating on the Karius test, that gives them an opportunity to also expand their reach.
Understand. So we've almost got a subspecialty discipline available within the healthcare system.
Yeah, for sure. And that's how we're evolving as an organization. I'm sure that's true with many other like-sized and bigger organizations.
Thank you. Uh, Back to Dr. Blauwkamp for a moment. Uh, we've heard some very compelling data from both from you and from Dr. Farrell about the essentially economic advantage of using the Karius test in a proper way with all the right guardrails. Is this a question that you frequently find yourself answering to prospective customers or systems?
I mean, this is one of the questions that comes up all the time, as you can
Right.
imagine, because in the end, we all have budgets and we need to figure out how to deploy newer technologies like this in the most responsible way. So the short answer is yes. Our efforts to address that thus far have focused on taking advantage of what we can learn in studies designed to prove out things like clinical validity. And so they're not the ideal studies to address health economic questions, for example, but we get the question so often that we'll often go in and do as good of a job as we can with a study like Pickup, for example, which was a study of patients with suspected pneumonia, where we're quantifying the cost of standard diagnostic workups and comparing that to the Karius test. And there's a paper on that, of course. Our future efforts, as we solidify that foundation of clinical validity across all these different applications, are shifting towards these clinical utility and these health economic outcomes. And so we've got a number of registries in flight now that are meant to address these with the right types of studies, the definitive answers that actually provide answers from how this test saves money at scale.
Thank you very much. And essentially the same question to you, Dr. Farrell, but with recognition that your system, like many, if not most systems, has an overwhelming number of Medicare and Medicaid patients within it, not necessarily always in private insurance. How have you dealt with this question from your own administrators, from insurers, or other parties who have a keen interest in the economics of it?
Not to mention, as a charity hospital, Bob, we also have
Yes,
a high number of air quotes self-pay population.
of
And
course.
the way I look at this, I mean, as I alluded during the presentation, I'm always leery about stepping outside. Well, in practice, I'm never bashful about stepping outside my lane, but when it gets into healthcare economics, it can get deep quick. And so I try and keep it simple, as I said in our take-home slides. To me, the question boils down to, is there somebody else who could benefit from fill in the blank? So in the example of the bronchoscopy, if I'm able to avoid tying up the interventional pulmonary lab and interventional pulmonologists time and space and staff, there's somebody else who's potentially benefiting. If on and then the next step obviously is just length of stay. That's the holy grail. If I'm able to diagnose, initiate treatment, and dispo the patient to home or wherever faster, is there somebody else who could benefit from that bed? And the answer is always absolutely.
That's very good. Dr. Farrell, thank you very much for that remark. And with that, I'll let you have the last word. I want to really commend both of our speakers and Karius for giving us a wonderful presentation. They've really opened a window on some new advances and frontiers in diagnostics with all the ready-made justifications and sense of stewardship that, of course, will be de rigueur now from now on. I'd also like to join you, Dr. Farrell, in recognizing Gary Prokoff. I recognize the disciple and chip off the old block, if he doesn't mind me referring to him in that way.
Well, I mean, I'm sure you know Gary, and he would take a chip off my block if I had not mentioned him.
I understand. I understand very
So
well.
I guess I would close not with a word, Bob, but 3 letters, my
Okay.
initials, JJF. So I really appreciate the opportunity and the invitation today. And I just want to have an open invite to anyone interested to follow up, continue the conversation. That's my email address, Bob, jjf@uic.edu. Feel
JJF,
feel free to drop me a question or drop me a line.
thank you very much.
You
And
bet.
so with that, let me go ahead and also add my best wishes to all of you in the audience. It is National Medical Laboratory Week. We've got hundreds and thousands of people who do this very important work every day, 24 hours a day, and we salute all of you. But I want to particularly now, as we close, I want to thank Dr. Tim Blauwkamp. I want to thank Dr. John Farrell. Obviously, I want to thank Karius and its laboratories. It was a great sponsorship I'm delighted that we've had a chance to present this information to all of you today. So finally, again, thanks to our speakers and thanks to Karius, and thanks to all of you who've taken a lot of time out of your day. You've stayed with us, and I hope it's been extremely interesting to you. We're getting a lot of kudos in the chat, for which I'm grateful, and I'm sure that Tim and John will feel the same way. So with that, let me bring today's webinar to a close. Thank you very much.